Hi everyone. The second part of lecture 12 is on cutaneous, which means of the skin. Mechanoreceptors, these are mechanically gated ion channel receptors, and they are going to help us with sensation of touch. So that's our topic for this section of the lecture. So mechanoreceptors are those that are going to respond to a physical force. So when we learned about the stereocilia on the hair cells in the ear, and when they physically bent, that pulled the ion channels open and allowed depolarization. Same thing here. So these are mechanically gated ion channels. You can see in the diagram here in the below image, you apply pressure and you can see it's pushing down on that cell membrane there, and that pulls the ion channel open so that now sodium or whatever can rush in and depolarize the cell. So for the cutaneous mechanoreceptors, there is a little difference from the hair cells. You might or might not remember that the hair cells didn't fire an action potential. They released glutamate, and they could do so in a graded response. Not so with the mechanoreceptors, so they're going to send an action potential. If enough of their channels get opened so that they get enough ion flow and depolarize sufficiently, they will reach threshold, they will send an action potential. All or none goes through the axon. So mechanoreceptors for the sense of touch. There are 4 different types that we find in the skin. So your skin has different layers, as you might remember from anatomy and physiology. The upper layer is the epidermis. Then the middle biggest layer is the dermis. And then you have the hypodermis or subcutaneous layer, which is where the fat is. So we have 2 types that are way up top here, not in the epidermis, but in the upper parts of the dermis. So we have these Merkel cells, which are also called tactile epithelial discs. So in anatomy and medicine, we're trying to get away from eponyms, which is where things are named after the person who discovered or first wrote about them, usually dead white men, and trying to just get into more descriptive terms. So it's a confusing time because you'll see both terms used. So I will not expect you to know one or the other. I'll always use both together so that if you know either one, then you will know what I'm talking about. So these Merkel cells or tactile epithelial discs are for very fine, precise touch information. When you're feeling the texture of an object and you feel the faintest little ridges in it, that you can thank your Merkel discs or your tactile epithelial discs for that. They're very good at discriminating very precise, minute differences in surface. So those are the Merkel discs. And then we have also located up here the Meissner corpuscles. They're called corpuscles because they have little wrappings of connective tissue around them. And they're really good for light touch, and they can also feel skin movement. So if you have something in your fingers and somebody's pulling on it, that kind of pulls the skin a little bit, and you can feel that. So these 2 are both for kind of some more precise things, but most especially the tactile epithelial discs or Merkel discs. All right, the other 2, moving kind of down through the layers of the skin, the next we're going to find are these Ruffini or bulbous corpuscles. And these are located in the middle of the dermis, and they feel stretching of the skin. So, if you pull the skin one way or another, they're going to sense that. So if you're getting a massage, you are getting lots of stimulation of your bulbous or Ruffini corpuscles, right? All that moving of the skin. If somebody's pinching your cheek, you're also getting a lot of signals from those as well. Then our deepest one is going to be the Pacinian or lamellated corpuscle. I have a student who remembered that it was lamellated because it looks kind of like a lollipop, so L for L. Lamellated also means many layers, and as you can see, it has many layers to it. These are good for detecting very deep pressure. As you can see, they're located deep in the skin, so you'd have to press relatively firmly in order to actually reach and activate these nerve endings, as well as vibration. So in addition to having 4 different types of mechanoreceptors in the skin, there are also different types of responsiveness. So the first one is slowly adapting, otherwise known as tonic. So if you touch my hand, then a slowly adapting or tonic nerve ending receptor will send signals for as long as you are touching my hand. Right? As long as it's there, it is sending signals saying it's there, it's there, it's still there, it's there. So I think they're very slowly adapting. They kind of never get over the fact that it's there. They have to keep telling you about it. That's how I remember it. And then the other type that we have are rapidly adapting or phasic. So they get used to it pretty quickly. So you touch my hand and you keep your finger there and they're like, okay, you're touching my hand, and then they kind of forget about it. And they're like, okay, whatever, it's there, but we're not going to send signals about it anymore. But then as soon as you take your finger off, they're like, oh, there's been a change. So they're really good at detecting phase change, right? So they're also called phasic. So they only send signals when they're a change. And we can see in the diagram here from the textbook, right, that these slowly adapting ones, as long as the stimulus is there, they're still firing action potentials, whereas the rapidly ones just tell me when something changes. So, oh, stimulus is here, oh, stimulus just left. So those are the differences between those two. And it just so happens that among those 4 different types of receptors, some are phasic and some are tonic. Am I going to make you memorize that? No. I just want you to know that some are one and some are the other, and that is going to explain some of the phenomena that we can see occurring in humans, like the fact that you haven't really thought about how your sock feels on your foot, except now I just brought your attention to it, right? So your phasic receptors, they gave up, right? After you put your sock on, they were like, okay, this is our new reality now. Yes, and they're not going to fire again until you take your sock off. The tonic ones keep sending signals. So you're like, so wait a minute, so why was I not aware of it until you mentioned it? Thalamus, right? So thalamus suppresses information where it's like, yeah, it's not important. Okay, I digress. The other important concept is this idea of receptive fields. So every sensory nerve ending has its kind of own catchment area that it is receiving information from. So in this example here, we see on the left what we would call a large receptive field. One axon from one sensory neuron is supplying this entire region of skin with its sensation, whether that be, Pacinian corpuscles, lamellated corpuscles, or Meissner's tactile corpuscles. There we go. Old habits, right? So it all just goes to one. So therefore, when that signal arrives, it travels through my spinal cord and up to the brain. I don't necessarily know if it came from this part of the receptive field or this part of the receptive field or this part of the receptive field. And so what they're showing you here, which I've kind of obscured, is this example of touching somebody with 2 different things at the same time. So this is called 2-point discrimination. So if I put 2 little pinpoint things and I touch you within the same receptive field, you won't be able to tell me if it's one thing or if it's 2 separate things, because you can't tell. So if this sounds familiar to like synaptic convergence when we talked about with the rods in the retina, right? Very similar, right? If it's all coming into one eventual signal, one retinal ganglion cell in that example, but one sensory neuron, first order neuron in this example, right, you can't tell exactly where it's from. Whereas if it's a small receptive field, so this example over here, right, I've got 2 different neurons. So I can tell the difference between something over here and something over there, because that's going to send signals on different axons. Now, if you touched me in 2 places within the same field, I would not know that it was 2 touches if it was at the same time. I wouldn't know it was 2 different objects. It would just feel like one. So it'll be fun to play around with that in lab. Receptive fields. Oh, Obviously, then, if you have lots of small receptive fields, you have lots of precise information, you have lots of axons. So the parts of our bodies that have very large areas of the cortex are getting lots of axons. They have small receptive fields. That's why they have such detailed information. The other thing is that for touch, for these cutaneous mechanoreceptors, these touch receptors in the skin, the types of axons that they all have are the A beta axons. So they're pretty fast. They're not the fastest, but they're pretty fast. So we're going to use some pretty fast axons for touch. And then let's talk about our three-step hop. So our first order neuron here is the sensory neuron. So this is our sensory neuron who, one end of it is the receptor, whether that be Merkel's discs, Meissner's corpuscles, Ruffini or bulbous corpuscles, or lamellated Pacinian corpuscles, whatever that end of that axon is. And then you're going to have the sensory neuron cell body sitting in the dorsal root ganglion, and then it's going to send, that axon is going to continue and go into the spinal cord. So that's our first order neuron. Now, you can see in the diagram below, which should look very familiar to you, that when it comes into the spinal cord, it's then going to start to go up, right? Because we're going to go up to the brain. And the part of the spinal cord that it's going to go up in is the dorsal column. So hopefully you remember that from our spinal cord lecture. So the dorsal column is this area of white matter here, right? And it's white because it's myelinated axons. These are the axons that are traveling up through the dorsal column. So here they go through the dorsal column. This diagram shows you here's some sensory input coming from down in the lumbar spinal cord. Here's some coming from the cervical spinal cord. And then they just keep adding to each other, right? As they're all going in the same direction, they're all going up toward the brain through the dorsal column, right? So through the dorsal column, these axons continue traveling upward. We're then going to reach the second order neuron. So we're going to synapse with our second order neuron, which is in the medulla for touch. It's in the medulla. Where is there it is. So we're in the medulla. And then as soon as we synapse in the medulla, which is happening here, and it's in different places depending on which part of the spinal cord you came into. Again, we're like a stickler for organization and everything in its place, but you don't need to memorize those names. But then it's going to cross over or decusate to the other side of the spinal cord. So after it synapses with the second order neuron, that second order neuron in the medulla is going to send its axon to the other side of the spinal cord. And that's what is called a decusation. And now we're on the contralateral side, we're on the other side, and we are going to travel, we were in the dorsal column, and now we're going to travel up through a structure called the medial lemniscus. I'm sure you've heard of it. Yeah. Okay, medial lemniscus. And that's going to take us to the thalamus. So here we are now, we've crossed to the contralateral side, we've decusated to the contralateral side, our axons are going up through the medial lemniscus, and then they're going to synapse with our third order neuron in the thalamus, which is then going to send nerve fibers out to the parietal cortex, to the primary somatosensory area. And that's how we get where we want to go. So overall, this is called the dorsal column medial lemniscus system. And I have a typo. I said metal. Olympics are on my mind. So medial lemniscus system, or the DCML for short. So let's just review. Our first order neuron, the cell body, is here in the dorsal root ganglion. It was receiving something from a mechanoreceptor in the skin. That signal travels along the axon and comes into the spinal cord and then goes up through the dorsal column until it reaches your second order neuron, which is in the medulla. Once you synapse with that second order neuron, it's going to cross or decusate, and then it goes up the other side through that medial lemniscus. Here it's traveling through the midbrain and keeps going. It doesn't get off at any exits in the midbrain, keeps going all the way up to the thalamus, where we're going to synapse with our third order neuron, which is then going to send axons to the somatosensory cortex in the parietal lobe. What about the trigeminal nerve? So again, we're going to have a ganglion, except this time we're not going to the spinal cord because we're a cranial nerve. Hello, we're a VIP nerve. We don't go through the spinal cord. We just go right into the brain. But we actually have a trigeminal ganglion. So it's the same type of structure, except instead of being outside of the spinal cord, the trigeminal ganglion is outside of the brainstem, outside of the pons specifically. So then we are going to synapse with a cell in the pons. That's our second order neuron. And look, we decusate, we cross, and then go up to the thalamus. And that's where we're going to find our third order neuron, which is then going to send this information out to the parietal cortex, to the corresponding region for the face. So in summary, our mechanoreceptors are mechanically gated ion channels. This keeps coming up. Do you think it will be on the midterm? I think so, right? And so they open in response to physical stimuli. We have 4 types that we find in the skin. The Merkels are tactile discs, which are for that very fine, detailed touch information. Meissners are tactile corpuscles, which also tell us about light touch. Ruffini or bulbous corpuscles, which tell us about stretching of the skin, and the lamellated or Pacinian corpuscles, which tell us about pressure and vibration. Some of these receptors are rapidly adapting. They get over it really quick. They only tell you when something changes. And others are slowly adapting, meaning as long as the stimulus is there, they're going to keep sending you a signal. Then we've got differences in the size of receptive fields, right? So how many individual sensory neurons are supplying any given region? The larger the receptive field, the less precise the information we're going to get. The smaller the receptive field, the more granular and detailed the information can be. Then our different pathways for the general body, it's the DCML or the dorsal column medial lemniscus pathway. So our first order neuron has its cell body in the dorsal root ganglion of the spinal cord. Its axon then travels up through the dorsal column all the way to the medulla where it's going to synapse. And then that axon decusates, goes to the other side, and then travels up through the medial lemniscus to the thalamus, where we synapse on our third order neuron, which then is going to send the signal to the somatosensory cortex. And in the case of the trigeminal nerve for the face, it's going to be the trigeminal ganglion, and then we synapse in the pons, decusate, and then go up to the thalamus from there. So that's it for touch. See you next for pain and temperature.